right now it's my very great pleasure to introduce Madeleine Beekman, who's a Professor Emerita at the University of Sydney, now lives in beautiful Far North Queensland. So Madeleine has been hugely influential in evolutionary biology of social insects, especially honeybees, but also enormously influential in bringing behavioural ecology into some less traditional complex systems like slime molds. I'm especially grateful to her for that personally, from rocking up as a fresh-faced young biochemist to the lab to start a PhD in honeybee genetics. As soon as I saw what Madeleine was doing with these crazy ants and slime molds, I was immediately hooked, and I've been doing the same sort of work. So, Madeleine, a very great thank you from me to you, and from us, you can speak to us today. So, welcome, Madeleine.
Because I have to start thanking Andy and Chris and Hoda for organising this meeting, which I think is extremely enjoyable. And I hope this talk is not going to disappoint, because it might be a little bit weird trying to combine a slime mold that has no brain, something that's got the biggest brain relative to body size, and that is us, through collective brains of ants and bees. And the main focus, I didn't try this out. The main focus will be on communication. So how do organisms that have these different brains, so brain is a shortcut for whatever they use to make decisions, how does communication fit in there? And in particular, I'm interested in how natural selection shapes the communication process of that particular organism in order for it to make decisions. And I'm doing that because I'm doing that because I'm using a very specific definition of intelligence. I basically go back to the root, the Latin root, which means interlegere. I've never done Latin, so I don't know how to pronounce it, which basically means the ability to choose between different options. Now, if you want to choose between different options, of course, you have to have information. So how do organisms with different kinds of decision-making processes gather information and use that information to make decisions? So you basically gather information about alternatives, use that information somehow, process that information, and then you make your decision. Now, so when we tend to think about brains, and of course this is the sort of brain that most people, not here, but most normal people, I would say, because they're not, to use the word normal, would think of is that a brain is a means to solve problems. So if you have a very complex brain, whatever that is, then that complex brain is capable of solving complex problems. But I would actually argue the opposite. I would say that it's the complexity of the problem that the organism needs to solve that has resulted in the complexity of the brain or whatever decision-making system it has evolved, because I am talking about slime molds and they don't have a brain. Now, hopefully I'm going to convince you of this. Oh, yes, and Joseph used the word environmental scaffolding in his talk. So the idea that it's actually the environment that drives problem solving. It doesn't matter, you can still read it. So this is my thesis: problems shape the brain and not vice versa. And all the problems that I'm going to talk about have a direct link to reproductive success. And that, of course, is why natural selection is such a strong force on these problem-solving abilities of these different brain systems. Now let's talk about when you don't have a brain. So this is the slime mold. The three people, one you know well, and he actually hasn't changed. I can't remember how old the photo is, but he hasn't changed, as you can see. So, working on slime molds keeps you young. The guy in the middle is Jules Smith-Ferguson. He came to me after he did honours with PGS in philosophy of biology. And he came to me and said, I want to work with my hands. I want to become an experimental biologist. So great, Jules. He was interested in slime mold, but he hadn't realized that doing experimental work actually requires a lot of work. It was a bit of a hard lesson for Jules. He found his place. He now works at the New South Wales Education Department, where he writes policy papers, apparently. And the other person there is Tanya Latti. Tanya came to our lab from Canada as a postdoc. And she's actually to blame for everything here on the slime-moulds because I was overseas doing field work overseas, and she sent me an email and she said, "Can I buy a slime-mould online?" I said, "Yeah, but have you checked the quarantine requirements or whatever?" Turned out you could buy the slime-mould for $20, which she did, and the world has never been the same again. Because that's when we started playing with this very strange organism. So why is it strange? So it looks like this when it's searching. So we call this the search front. And what it is, it really is a bag of slime. It's a unicellular organism. It's unicellular because even though the nuclei in a mitochondria divide, like in any normal cell division, the cells don't divide. So it just gets bigger and bigger and bigger. And if you keep feeding it and giving it space, it's unlimited. We once wanted to do an experiment how big we could grow it, but we gave up because it needed too much space. So these veins here is what it uses to shuttle cytoplasm through its body, if you call it a body. And once it found food, so this search front, it will bump into food and then it will completely remodel its body, and it will make those big veins connecting the different parts of its body to the food source, and it can then also exploit multiple food sources at the same time, and it makes very efficient networks. So collaborators of mine, before I knew them, and many people actually in the room know about that work, they used the slime-moulds to solve mazes. They did 2 particular experiments, and for both of them, they won the Ig Nobel Prize, which of course is for science. It makes you laugh first and think second. And in both instances, they sang their acceptance speech. They're Japanese, and a lot. Now, what we've shown, and other people too, is that despite not having any central nervous system or anything that looks like a central nervous system, this thing can habituate, which is known as primitive learning. So it can overcome a negative stimulus once it has learned that if it overcomes that stimulus, it will be rewarded with food. It can make trade-offs during foraging, as PGS mentioned yesterday morning. It can behave flexibly during foraging, which means that if it's already exploiting one particular food source and it then finds another one that's better, it can rearrange its body and change its focus to the more profitable food source. It can behave irrationally, but it behaves irrationally in an economic sense. This sounds extremely weird. This is a particular experimental paradigm that is used in humans, stickleback bees, and the idea was always that it's a complicated story, and it requires a very specific experimental setup. But our idea was that this is due to too much information, and that the brain can't process all that information. And then our idea, or Tanya and my idea, was that if you don't have a brain, you can't get confused. Well, lo and behold, the slime-mould also gets confused, whatever that means. As I said, it's a very specific experimental setup, but it was very fun to show that something that doesn't have a brain behaves in the same way as humans, given the same question, basically. And this is work that Chris has done. It uses an external memory, sort of in the same way as Hansel and Gretel use breadcrumbs to find the way back to home, and the evil stepdads try to get rid of them by dumping them in the forest. But the slime-mould uses it differently. It uses the leftover of these veins. So these markings are left when it has moved over the environment, and then it knows that there's no reason to go back again because I've been there, so I've probably eaten everything that was there. So it actually uses it as a marker to know that it shouldn't go back. Now, so that was all a lot of fun, but if you're interested in learning, etc., or how brains work, then apparently associative learning is the holy grail if your organism can do associative learning, then it's sort of a step up in the cognition step, say. Now, but also with a large number of other people, have tried to figure out if slime mold can make associations, which would be quite remarkable. As I said, it has no neurons, and it's hard to envision how it could store information and compartmentalize that information. So this is the setup that Jules used. So the idea was to always have an association between a negative, or not a negative, it's supposed to be a neutral odor, lavender, which I know is a complex stuff, odor. It's not a single component, but for some reason we decided to use lavender. And the idea was that if you constantly, or in 2, 4, and 6 trials, constantly make this association, can the slime mold learn that when it smells, or when it perceives lavender, that it's always followed by food. So this is a very easy setup, simple setup. So it starts at the top, then it bumps into food, then lavender, then food again, or the other way around. Now, because it's pretty difficult when you work on an organism that is totally different from what you are, because you have to understand what it can perceive or not. And that's why many of the other studies that have tried to find associative learning could be criticized for saying, well, maybe the negative results are because it can't perceive what you think it can perceive. But we're pretty sure that it can perceive these odors. So what you then do is then you test it in a Y maze. So you give it a choice between lavender and nothing. And of course, if it had made the association, then it should always go to the lavender. And it probably won't surprise you that that's not the case at all. So these are the results of the Y maze choice. And you can actually see the blue is where it shows lavender, that it has it's actually slightly repellent, which really shouldn't matter for the experiment, because if it really can make associations, then it should just go to the lavender and overcome its negative feelings for the lavender, because it can habituate to negative stimuli. So we had finished our explorations in the slime mold world. So it cannot store information to create an internal memory. And I have to admit, so that was for us, it was sort of the end. Moved on, let's move on. I know Chris has done more fun work that you may talk about later, not sure. Anyway, so the slime mold work hasn't totally ceased. Okay, so let's move on from no brain to a collective brain. I'm going to talk about ants very briefly and a bit longer about bees. So as I said, my interest has always been in communication. So how is the communication system shaped through natural selection to allow the organisms to exploit their environment optimally, or whatever optimally is. And let's start with ants. So you may think of ants something like this. So the trail-laying ants that you find in your kitchen when you were too lazy to clean up your spilled cake again. But this is actually only one type of ant, the one that has really large colonies and they form those pheromone trails. But there's actually a huge variety in colony size, which is not surprising because ants are one of the biggest but in group of species on the planet in biomass. So we actually have some species that only have 3 individuals in a colony, and then up to the ones you'd probably think of, several millions, for example, the army ants in Africa, South America always mix up which are the biggest. Now, colony size definitely shapes the need, because you can imagine if you have a large family, you have more mouths to feed, and the same is true for the ants. And we heard about tandem running again in Joseph's talk on Tuesday. So this is where you have small colonies. He showed Temnothorax, which is one of those small colony species. They have a few tens of individuals. And when a forager has found food, she will basically go back and entice another individual to follow her, and we call that tandem running. So there's very direct communication, and I'm now seeing that I've got the wrong slide up. So I'm so often ahead of myself. So if you only have 3 individuals, you don't have to communicate at all because there's only a few mouths to feed. So this is actually what I'm talking about now, tandem running. So this is the first form of direct communication. And some people have said that this is a form of teaching. Other people have criticized that for a reason that I can't remember. But it's a one-on-one relationship. Now, if colony size then becomes a little bigger again, we see a combination of direct and indirect recruitment. So here we have the same aunt again, but now she goes home and takes a group of individuals and guides that group to the forage site. And she can also then use some pheromones to make it easier, because now you don't necessarily have the direct contact between the recruiter and the recruitee. And then using the pheromone makes it a little bit more efficient. Now then the ants in your kitchen, they are the mass recruiters, and they rely solely on indirect communication via that pheromone trail. When I started working on ants, I had the idea, or trail forming ants, I should say, I had this vision in my head that this is an extremely inflexible method of communication, because the pheromone can stay for longer than the ephemeral food source. And I could just see that if the food source is no longer available because they've overexploited it, the pheromone will still be there and individuals will be stuck in a suboptimal solution. Now, mass recruitment is really good in quickly getting a lot of individuals to forage on one particular site, which is important if you live in an environment full of other competitors. And also when your colony is so big, you really have to be as efficient as possible. Now, it turns out in large measure to work that Chris has done is that this mass recruitment system is actually much more sophisticated, because individual ants can adopt or adapt their pheromone laying depending on the circumstances. So it's definitely not as rich as I used to think. So different kinds of communication depending on how essential it is for the colony to quickly gather a large amount of resources. All right, now let's talk about bees. Bees moving home in particular. Now you think this is a shitty video. It will be. But what you're looking at is a swarm of honeybees and the bees are trying to figure out where on earth they're supposed to be going. So is the person who's filming them. Because he's actually in the swarm and trying to figure out what's the direction that they're going to fly into. Now, this is an extremely tricky problem for bees. So it has everything to do with nest site selection in the genus Apis. And I'm not just talking about Apis mellifera, the western honeybee we have here, but I'm talking about all species of Apis. And there are 11 or 12, depending on whom you ask. And most of them live in Southeast Asia. And they're faced with a problem that looks like this. So the details matter for these different species, for the biology of the species, but there's one thing that's the same. So when they reproduce, they make what we call a reproductive swarm. In the middle there somewhere is a single queen, and the swarm now needs to find out where they're going to move to. So they've left their old nest, and this is basically the daughter colony with the old queen, which is a bit funny. And they're extremely vulnerable because you can imagine heavy rain, a storm, predators, and the swarm is gone, and therefore the reproductive output of that colony is also gone. So they have to make a decision on where they're going to move to, but that decision has to be quick. But depending on how important it is where they're going to move to, not so quick that they make a bad decision. And I've spent years, again, working on nest site selection in different species of Apis, and this is arranged, just a few people are relevant for this particular part. I was going to look up, so that's Konrad Diewald. He was a PhD student in computer science in Leipzig, and I was going to look up what he's doing now, but I forgot, I got distracted, so I don't know what he's up to now. Given that he did computer science, he's probably doing quite well. This weird person is Tim Scharf. He was a postdoc in our lab, and he's now at the University of New England. James Mackinson He's the guy who was trying to find out where the bees were flying, did his PhD, obsessed with bees, and is now at the University of Western Sydney. And Ben Aldroyd's been my partner in crime for a very long time in all different ways. Now we're planting trees in far north Queensland, where we spent years and years and years in the field trying to understand what the bees are doing. So the way our approach has always been, you study bees, you mark them individually, you try and figure out how the individual behavior affects the collective behavior. And then with the help of people like Conrad, simulations, and Tim, mathematical biology or applied mathematics, you then try to see if this idea you have about the rules that the individual bees use, can that really be translated in the collective behavior as we see it? And that's been an enormous a lot of fun. Some people want to stay inside. I prefer to be in the field with the bees. And so that works really well. So this is the basic problem that the bees need to solve. So they need a single solution because they have a single queen. But of course, the environment is replete with potential nest sites. So how do they narrow it down? It's the best out of n problem. So how do they find the best solution out of all potential solutions? It's a two-step process. You have to select a new nest site first, which is done by only a subset of the bees. We call them the scout bees. They come back to the swarm and they dance. Talk about that later. And then there's the guidance of the flying swarm. Now, the two processes are linked because you can't guide someone if you don't know where you're going. At least I wouldn't recommend it. It's not a good strategy. So the two processes, selection and guidance, are tightly linked. So that means it's the same bees that are involved in both processes. They're different bees and they have different needs when it comes to nest site selection. So these are the three big groups of bees, the cavity nesters. So that's the bee we have here, Apis mellifera. But this is actually a photo of the Asian hive bee, Apis cerana, that found a hollow Buddha and uses Buddha's mouth as its nest entrance. And it nicely illustrates the difficulty of the problem for this bee because it needs to find a hollow tree, its normal nest site, and the entrance can be this small in one tree in the forest. So that's a pretty tricky problem. The alternative are the dwarf bees. So this is Apis florea. It makes a comb that's about this small, a single comb, and they can nest almost anywhere. They just need some shade and they need to be able to protect themselves against bloody green tree ants, which want to eat them. I don't like green tree ants. So, and they don't either. And these are the gregarious nesters. They are the giant honeybees. They are big, they're about this big, but it's the comb that gives them the name. So the combs can be this big, this long, and they nest in big trees because otherwise the tree can't hold their weight. And in Southeast Asia, you often see them on big structures like water towers and temples, etc. They're gregarious nesters. They're also migratory, so they follow the flowers and they come back to the same place, and no one knows why. And no one knows how, because the idea is that the bees that were there before are no longer alive by the time they come back. We tried to solve that problem, but failed because they're really difficult bees to work with. I'm not going to talk about them. I'm going to talk about the first two, the laissez-faire ones and the I really want to find the best nest site one. They all use the same communication, and that is to dance. So they use this the waggle dance, and it has 3 pieces of information. And this, of course, is for foraging. The direction in which they need to fly to relative to the sun, the distance they need to fly in, and how profitable the source is that they have found. So it's not a lot of information, but it's pretty helpful information. Now, because the two processes are linked, it's only the bees that dance just before the swarm lifts off that will attempt to guide the swarm. So that's important to remember. But the dance is not as precise as you may think it could be. So these are 2 plots with 3 bees, and they're individually marked. So this stands for light blue, purple, purple. Yes, we have a lot of fun putting paint dots on bees. And this is probably also Apis florea, and they're tiny bees. It's a lot of patience. But so if you look at the same symbol, you can see that the information encoded in the dance as we extract it from the dance is quite messy. So it's not that I'm pointing, well, the supermarket is when you go out of this building and you turn right and you turn right again, it's there. No, it's, there. Go and buy milk, bearish. And this is, well, for one thing, they don't necessarily have to be that precise, but also they can't be more precise than they are. And I can read the top. So, as I said, we spend enormous amounts of time looking at how bees behave and then try to understand how that translates to the swarm guidance in this particular case. And when we're working on Apis florea, which is the bee that can just basically nest wherever it can, we thought that this is probably the process that they use, because what we noticed just before the swarm lifts off, there are lots of bees still dancing in different directions, not 360-degree directions, but it was narrowed down to something like 60 degrees. And we thought, well, is that enough for the swarm to be guided to where they want to be? So we came up with this idea that for every dance, you can calculate the directional vector. You can add them up, and then you get this purple line, which is the vectorial consensus. And the idea was then, which we then tested and turned out to be true, that what they're aiming for is a consensus vector. So as I said, they basically need to narrow down the options in 360 degrees to something more akin to about 60. Then they'll lift off, and everyone who was dancing prior to liftoff will still try to guide them in the direction they were flying to. And somehow they manage. They just get somewhere, and once they get to a stand of trees or something, they make up their minds there then, and then they settle, which is a different process again. So very laissez-faire and very quick, because the decision doesn't have to be very precise. On the other hand, we're talking about Apis mellifera, so the one that nests in the hollows. The nest site has to be advertised really precisely, and that means that only bees can guide the swarm when they all agree on where they're going. And that's a very complicated and beautiful process, which I don't have time to explain now, but they have a very intricate process that leads to a quorum. So all the bees, it's not consensus, they all agree this is the place we are going. So that means that all the bees that fly off guide the swarm to the same place. And we've done all sorts of wonderful experiments by making them really confused and show that then the swarm cannot be guided. So we then tested this in a computer simulation, and we call it a tunable algorithm, but basically the same. The algorithm is the same, but it's fine-tuned by the difficulty of the problem that the bees need to solve. And this is probably not very helpful, but I put it up anyway. But it was inspired by looking at the bees, coming up with a theory, testing the theory, and then testing the theory back in the field. Which I think, well, I'm biased, but I think that's the way one should do things. Okay, so natural selection has shaped the decision-making process of these bees. If the problem is really difficult, then the communication system becomes much more complex, because otherwise it can't solve the problem. Problem first, natural selection acts on the ability to solve the problem. And that's how we get these complicated decision-making processes. And now, something completely different. So from the collective brain, our brain, collective brain, we move on to the vertebrate brain, or one brain in particular. So this is the question I fell into. I didn't set out doing this, I just fell into this. So why is our brain so large? Now, given the way I set it up, then of course the problem, or the question I need to answer now is, what is the problem that has shaped our brain? Why has our brain become so enormously big? And of course, it's big relative to body size. Don't forget that. Now, I'm going to argue that it is the direct or indirect result of a decision, an evolutionary decision, that our early ancestors made about 4 to 3 million years ago. So this is maybe going to be a little bit confusing when I talk about humans, because when I talk about our species, I'll specify, but when I talk about humans, I'm talking about a long line of ancestors ever since the human lineage splits from the common ancestor of chimpanzees and bonobos, which is about 6 million years ago. So about 4 to 3 million years ago, our early ancestors decided they were becoming bipedal. So it came down from the trees, and nothing has ever been the same since. Are there any runners in the audience? Are you sprinters?
How far in history can we go back?
I used to be a sprinter too, but my legs are too short. But can you two outrun a 4-footed organism, you think, at your fastest speed?
Yes.
Really?
Yeah.
We put it to the test.
Yeah.
I mean, I'm talking about a dog.
It was a very old dog.
Yeah, I know. In its prime. In its prime. I bet you can't, because we gave up a lot by becoming bipedal. So not being able to run very fast. Yeah, we have Gad, who is of course now our Queensland icon, this young chap who can run very fast for a very short period of time, but he's not normal. I think we can say he's not normal. So we gave up this ability to run fast, and that in a surrounding that is filled with extremely dangerous predators. So these are only 3 pictures I could find when I looked for big predators and that lived in Africa 4 to 3 million years ago. But you get the picture. So here we have, because now we're talking about Australopithecus afarensis, Lucy, better known as Lucy, because I'm short, but she was probably shorter. No weapons, no claws, no big teeth, not being able to run fast. So that's a huge problem. So the only thing that kept our early ancestors alive was relative safety in numbers. So this is when our social life began. Of course, it may have already begun up in the trees, but then by coming down from the trees, that has just put an enormous selective pressure on becoming social. But there are more reasons. If you only have 2 legs and you're silly enough to break one, as I managed to do a few years ago, you become completely dependent on other individuals. So here's my little nephew who really wanted to push my wheelchair, which was going just fine until my brother, who was there, joked to his son when we were going down the slope, "Now just let go." Whatever. Did exactly that. So I broke the other, no idea. Now, you can say this is a new invention, but we know of at least one fossil, which is almost 2 million years old, that had evidence of a broken bone that had mended, a broken ankle. So that means that this individual, while he or she could not walk, was looked after by other individuals. So again, it's another piece of information or piece of indirect evidence that our early ancestors were already social. And there's another issue. If you walk upright, you can't chuck your baby on your back like this gorilla mum did, because even if you would still be very hairy, which they probably weren't, the baby can't hold on to an individual that stands upright. So babies literally became a handful. And now you're holding babies in your arm. What else can you do with your arms? Not much. So again, you become very dependent on other individuals around you. So all these problems that came from our ancestors deciding the trees weren't fun anymore, we're going to walk on the savannah, started this, shaping our brain, I should say, to become highly communicative And I'm talking about non-verbal communication. I'm talking about things like empathy, mind reading, understanding what other people are saying, a lot of emotions. I mean, Charles Darwin wrote about emotions, how we can still see emotions in faces. And that's not a novel invention. That is very old. So this is how our brain started to become geared towards communication. Let me just check what the next one is. Now, that was probably the first driver of brain size evolution. So this creature, I forgot the first Sahelanthropus, I think, still lived up in the trees. This is Lucy and her ilk came down. So that's the first occurrence of bipedalism. And you can see that there's a steady increase in brain size of the different species. When one species morphs into another is a huge debate. So you can't actually really appreciate how bigger our brain is relative to the others, because here they group Neanderthals and Sapiens, and they really should be separated. So there's a steady increase in brain size. Now there's one species that I want to focus on, and that is Homo erectus. So Homo erectus had been around for a long time. It probably made its first appearance almost 2 million years ago, went extinct about 110,000 years ago. So keep that in mind. Homo erectus was still around when Homo sapiens evolved. Now, within that period, brain size within that species increased tremendously. But to explain why, I first have to distract you with our beloved royal family. So here we have two princes. One is the heir to the throne, and the other one is Despair. He even claims to have written a book that's called Despair. Now, why is this important or interesting? The heir to the throne is very restricted in what he can do, because they mostly were his. His function is basically very constrained, because after all, he has to become the future king. Despair, on the other hand, has much more freedom, which of course Harry has been exploited by foul-mouthing his family so that he can become even richer. So Despair has a lot of leeway, and it can do things that the heir cannot do. Now, why is this relevant? It's only relevant because it's a nice metaphor for what happens when we have gene duplication. So gene duplication is an extremely strong force in evolution, not just in our species, but in all species. And I'll talk about two influential gene duplications in our species. So here we have the original gene that, of course, performs a particular function, and that function is important, otherwise the gene wouldn't be there. Then for one reason or another, it gets duplicated. So now we have the heir, still needs to do the normal function, and the spare. Now the spare can do one of two things. It can do nothing, in which case it probably drifts out of the population, or it can do something new. And this is the innovative power of natural selection. So when there's a gene duplication, it could work on that duplicated gene and give it a different function. Now there's two that I want to talk about in our evolutionary history. A gene that made us sweat, AQP7, and a gene that made our brain balloon, NOX2NL. And you can see that people who name genes have a real creative mind. So the first one, aquaporin 7, it's one of the gene family, so that's why it's got a 7. We have 5 copies. And this gene does, or the fact that we have 5 copies gives us 2 traits. One is to sweat profusely, which when you live in far North Queensland is not always handy, but it's essential because otherwise your brain basically cools. So most of the sweat glands are in our head. It is unique to humans to have so many sweat glands, and it really keeps the head cool. So the brain is extremely vulnerable to overheating. That's why fever is so potentially dangerous, and having the sweat glands on the head allows us to keep a cool head, particularly during exercise, because that's a dual function of this aquaporin gene. It allows the quick release of energy. So now you can run for a long time, because you have the energy, and you can keep doing it because your head is not going to overheat. Now, this probably already occurred in Homo erectus, and Homo erectus therefore became a long-distance runner. And it literally ran out of Africa. It's the first species of Homo to have left Africa and spawn a lot of different species in Europe and in Asia. Now, that's not of interest to me now. Of interest to me is two things. Well, first off, because I started asking the question, why did it increase its brain size? It could increase its brain size because of the evolution of endurance running. So this means that you can outrun your prey. So now Homo erectus no longer had to depend on scavenging because it couldn't kill anything bigger than itself. But it could outrun its prey, because if you run after an antelope, and they were not sprinters, they were endurance runners, so it's a slow, steady pace. The antelope is just chased, and the antelope at some stage has to rest because otherwise its brain would cook because it doesn't have the sweat glands, and then you can kill it. So the inclusion of a large amount of meat in the diet of Homo erectus, and this is a vegetarian saying this, has been essential in our evolution, because it allowed Homo erectus to have a larger brain. Because the brain is the most expensive organ that we have, and in particular, a big brain costs an enormous amount of energy. So there are other things related to this running. The skeleton changed because Lucy, yes, she could walk on two legs, but she could definitely not run. Now, I've been told that we have inherited the skeleton of Homo erectus. So apparently, if you look from the neck down, you can't easily distinguish between a Homo erectus skeleton and a Homo sapiens skeleton. Above the neck is a different matter. So the pelvis in particular changed to allow this running. So two things needed to be done. The center of mass needed to be lowered to increase balance, which basically changed the shape of the pelvis. And this is important because the birth canal also changed, which will become important later on. And also we now have these hip bones, the iliac bones, which stick out much more. So sorry, I'm showing the chimpanzee and a human. And the iliac bones were required. They had to become bigger and flare out to form a better attachment of the gluteal muscles. And the gluteal muscles are basically what you need to run. Walking is a completely different thing, but for running, you need a big bum. And that just means that your bum is for running and not for sitting. Now, this is all fine. So we have a beautiful skeleton adapted to endurance running. Its brain has increased because it's now eating all the poor little antelopes that it killed. And that was all fine until in our species, and I'm talking about Homo sapiens, something weird happens. So a formerly broken gene repaired itself and then made copies. And that is a curious case of NOX2NL. And there are probably neuroscientists in the room who are going to be abhorred by my very shallow explanation for how we got so many neurons. But I'm an evolutionary biologist, so that's my excuse. So the idea is here. So in our ancestors, relatives, the gorilla and the chimpanzee, there is what they call the pseudogene, which was a duplication of this NOX2 gene, but it has no function. So it just sits there and does nothing. And somewhere in ancient humans, that gene, the pseudogene, became a real gene. It started to have a function for one reason or another, and then it got duplicated. There were two duplication events because we all have 3 copies of what became known as NOX2NL. Because I also have to say that when you look at Homo sapiens, old specimens of Homo sapiens, call them ancient humans or archaic humans, their brain size is still much smaller than the brain size we have now of modern humans. So somewhere that duplication happened. Now, why is that important? So neurons, they start by, say, the neuronal stem cells or neural glial cells. Those cells can either duplicate themselves or they clone themselves, so you get more radial glial cells. But at some stage, they start differentiating into neurons. And that's a one-way street. So once they start differentiating, they can no longer make more glial cells. Okay, so glial cells turn into neurons via a large number of other cells. Now, the ancestral expression of Notch2 and L results in a quick differentiation into neurons. Now, this new version and the duplication basically delayed differentiation into neurons. So what you now have is at the base of your brain, these glial cells keep copying and keep copying and keep copying and keep copying and keep copying, and then very late they start differentiating into neurons. And that then results in the ballooning brain, which I'll get back to. Now, it's also important to note that this is really living on the edge because, as I said, we need 3 copies for a brain development. If you have 2 copies, you suffer from microcephaly. If you have 5 copies or 4 copies, you suffer from macrocephaly, and both of them are associated with mental and brain disorders. So this is really natural selection putting us on the knife's edge. This side's bad, that side's bad. So we need to stay on there. Okay, so this is just basically what I'm saying. So this is where this process happens. Then they migrate to the neural cortex, and that then results in this cartoon that I showed you earlier, which is quite good illustration. So now we get the ballooning brain. Now remember those narrow hips and what I said about the narrow pelvis and the change in the birth canal. So we now have our species now has the skeleton of Homo erectus for the brain of Homo sapiens. And that doesn't work. It doesn't work for females because now during pregnancy, a mother is nurturing a child with an extremely expensive organ, the brain, that needs to grow, for which she needs to be able to produce a lot of energy, enough energy to grow that brain and keep herself alive. But also, if that brain gets too big, it can't fit through the birth canal. So that means that ever since we had the gene duplication and the big brain, babies needed to be born extremely prematurely. And I call that they're seriously underbaked. Now, the criticism I've had is that people say, well, many species have very underbaked babies. And yes, it is true, but you can just look at the details. When a human baby is born, their brain requires 80% or 85% of their basal metabolic rate just to keep that brain going. If something goes wrong, it can't get mother milk quickly, for example, it'll die. When a chimpanzee's baby is born, I think their brain is about 40 or 45% of the adult weight, mature brain size, but also it uses a fraction of the energy. So our babies are special. They really are seriously underbaked. And people have done studies on extant hunter-gatherers to try and see at what stage in a child's life is it capable of gathering enough resources to sustain itself. And that calculation came out in about 15 years. So for 15 years, an average human child needs other individuals to take care of it. Now, if you have teenagers, you're going to say that 15 years is total underestimate, but this is a very particular way of measuring it. So I call that our original childcare problem. So this is the big head of the baby, and that caused all the problems combined with this skeleton that we inherited from our ancestors. But in a twist that only Mother Nature could have come up with, the cause of our original childcare problem, this big head, also provided the solution. And that solution came through the remodeling of the head. Now, you can think of the skull as one big structure, but it's not. It's very modular, and it can change quickly to adapt to this increase in grey matter. So our head became very different. So you can pick up a skull from an archaic human and a modern human, and they look very different because we have this weird bulbous head and a very flat face, and that has everything to do with the need to change the shape of the skull in order to fit in that brain. Now, we've already heard about the descent of the larynx that gives the low voice. So that was a consequence of the change in the shape of the head and the neck, but it also pulled down the tongue. Now, why is that important? You could see this descent of the larynx as a design fault. And actually, when I started looking into human evolution, I came to realize that our evolutionary history, and of course this is true for all species, every time something silly happened in our evolutionary history, species basically had two choices: adapt or die. Now, we adapted because otherwise I wouldn't be talking to you. But the descent of the larynx is one of those weird flukes. So it's good in a way because it increases the size of the oral cavity, which is important for what I'm going to talk about next. And it increased the control over the tongue. I hadn't realized this, but half of our tongue sits actually in our throat. And that gives us very precise control over its movement. But it also, the descent of the larynx, also increased the risk of choking. And apparently, which is a bit scary, choking ranks as the first leading cause of unintentional death globally. So this is a huge cost. So you would think this design fault should have been selected again from day one. Everyone's choking, that's the end of it. That is not the case. And the argument is that the benefit of the descent of the larynx, with all the other modifications that came with it, allowed one thing, and that thing is uniquely human, the evolution of speech. So what do you need for speech? You need control over breathing. You need, and control over breathing is important for phonation because the breathing allows you to change the air that flows off your vocal cords. Then you need to articulate, for which you need control over your tongue and your lips. And all that is orchestrated by the brain. Now, we had this amazing brain because our babies are born. So first off, we already had a brain that was geared towards communication, non-verbal communication, because of the need to be social. Then we created these insanely immature babies who are born with an extremely plastic brain. And all these things together geared our brain for the ability to allow speech once we got the hardware to do so. So this is basically what I'm arguing. So the origin of our weirdly large brain, everything to do with our ancestors' decision to come down from the trees and to become bipedal. Because of the need to be social in order to be alive, being bipedal in this environment, having your hands full of babies, needing others to look after you when you broke your leg, that provided us with the software for more precise communication through sociality. Then this remodeling of the head gave us the hardware, the ability to control our breathing, or not through our breathing, but the ability to use our mouth and our tongue to make precise sounds as we control our breathing, and therefore the way air flows over the vocal cords. So I would see it as a manipulating brain, which is all the baby's fault. So it's not their fault that they're born so prematurely, but they are born with an extremely plastic brain that turns them into learning machines. And of course, they have to be learning machines because if no one looks after them from day one, they are dead. So they really depend on all these individuals to look after them for more than 15 years. So that vulnerability turns them into master manipulators. Anyone who has a child knows exactly what I'm talking about. And the parents can't do it themselves because it takes so long to raise a human child. The reason why we managed to deal with this is because of the origin of language. Now, this is something I don't have time to talk about, but when I realized that when our species came into existence, there were about 5 or 6 different species of Homo sharing the earth with us, which I just find amazing. So you walk into a lecture theatre and you don't just see Homo sapiens, but you see all these other species, potentially. But of course, in no time after we came into existence, they went extinct. Because one thing that we could do with this amazing ability to talk, we could rapidly increase our reproductive rate. Because it's very counterintuitive. We have the most expensive babies, but we make a lot of them in really quick succession. So as a chimpanzee would take about 4 years before it has a second child. In humans, and of course when I'm talking about humans who lived as hunter-gatherers, that interval is about 2 years. So you have a tiny toddler, and then you have the next one, and 2 years later you have another one. So that's an enormous increase in reproductive rate, which is only possible through speaking, to ask for help, and list grandmothers, because menopause is another thing that can be explained through this. And now we are alone as humans. Okay, so Charles Darwin also thought there was a feedback loop for why we have such a large brain, but his was quite different. So he thought our hands became free by being bipedal. That allowed us to make tools. And by making tools, we could now outwit our predators, not through strength, but by being clever. Now, that then meant that if you were slightly more clever by having a slightly larger brain, then you had better problem-solving abilities, could make better tools, and that was the feedback loop that led to our large brain. Well, I've done it, I think it's differently. He was right. Bipedalism is essential. By becoming bipedal, we had to be social, which led to high cognitive abilities and skills. Then I leave a lot out here, but then we got the exceptionally needy babies for our species and the gene duplication, further increase of cognitive skills, and basically babies are the driver of our large brain. So the problem shapes the brain, as I said. Slime-mold has a very easy life. It just crawls through the environment. There's plenty of food. It only needs to make decisions about where to forage and when. No brain required. For the ants, hopefully I convince you that if you need a lot of food really quickly, then you have to have a much more complicated decision-making or communication strategy to allow the colony to thrive. Bees, if you have a very difficult problem, again, you have to fine-tune the communication channels in order to solve the problem. And then we have the biggest brain of all and one of the biggest problems. And if you're not convinced, then you should read this book. And if you are convinced, you should still read the book. Because there's much more in the book than I had time to talk about. And also I followed up some of my ideas after the book was published on this Substack. So it helps me if people read what I wrote. Is it better read than dead? And that's definitely true. So this is a lot of work that I covered, which I've done with a large number of people. Some of them I've mentioned, not all. And also, of course, the World Templeton now fell off. And I've been really lucky in my career that I've always had a lot of money. And yes, this little child is related to me, and it's all her fault. And thank you for listening. Realized I was a bit. Once I get going,
Thanks so much. Any questions from the room?
I'm still thinking about that dog, are you?
I am. So Madeleine, I want to explore and just push the hypothesis a bit. So it's that you came out of trees, bipedalism. Problems are solved that shape the initial brain evolution are cooperative care and cooperative defense. Yeah.
Well, the other way around. Yeah.
No, we get bipedalism,
Yes.
cooperative care, cooperative defense, brain growth, brain upgrades, right?
Initially.
So what I'm thinking is, why didn't we become social? So we've got the social animals, they're solving the problems of cooperative defense, cooperative home care, social, they're communicating. In all the cases, other than the primates, that use social innovations accompanied with a shrinking of the brain of the individual. So why in primates did the solution of those problems cause a brain malformation? And we know that eusociality is possible in mammals because
Our brain has been decreasing apparently. And we're not used social in the sense of, so we do have alloparental care, although people call it differently because it's not actually younger individuals looking after the babies, but it's what I was talking about. The evolution of menopause is one other aspect which has been important in our evolution. And I can't remember the word that they use, so it's not alloparental care. Why have we not become?
I guess my point is there's solutions to cooperative care, cooperative defense, and communication and sociality that aren't accompanied by brain growth. So the question is, in that initial stage, why in the primate line was it accompanied by brain?
I guess because different things happen in evolutionary lineages, I would say. Because the vertebrate brain is very different to an insect brain, for one thing. And I don't think we could increase our reproductive rate to the same extent as even a mole rat. So the brain has already been big, and I don't think you can easily shrink a brain once you have a large brain. That might be another constraint. I think you can go extinct if it's too big. But I mean, primates all have a relatively large brain. So I think it's something of the whole primate lineage. But it does seem to be the case that our brain has shrunk.
I'll just throw maybe potential answer question. Could it be because of the need for an infertile caste that you actually see in a eusocial group?
Well, that's why I was saying, not just an infertile caste, but also a super fertile caste. And I think that's probably the main constraint, because you can make eunuchs. I mean, they've done it in the past, making people sterile, but not in an evolutionary sense. But I think there's no way primates could increase their reproductive success to make it worthwhile to have a large number of individuals who don't reproduce at all. Thank you for helping me.
Notch is a really good example, actually.
What is, sorry?
Notch is a really good example of one of the strongly evolutionary perspectives that we're trying to get back. So notch in the inner ear stop reproducing inner ear cells. That's why you go deaf. But we procreate before we go deaf, so there's no evolution. Correct. Birds regenerate their inner ear all the time because they need to be able to. They're noisy environments, but they lose their hearing, but they keep regenerating it because they need over the period they go deaf. It's fundamental shift in something like notch signaling. And we spent a lot of time trying to see where you could reverse engineer that to get hearing back. So it's a fundamental pathway that so strongly evolved in communication as well.
Yeah, and once you've been on that track, you can't go back.
Can't go back.
Or you go extinct, as you
Yeah.
said. Yeah. Either you deal with it or you can't.
Could you go back to your last slide or the previous one? Yeah, there. Yeah, this one. Bipedalism are early ancestor social. I would say socially dependent. Because the ancestors, they were already social. And I think that might be why the brain did not shrink. Because you become socially dependent on others for your survival. And for that, you are going to need, what you mentioned, theory of mind and all the other cognitive machinery that will come later. And I don't think that you can do that with shrinking the brain. Is quite the opposite.
Yeah, thank you. I'm glad that other people can answer your question.
Elena, what do you think about the social brain hypothesis? Consistent, what you're saying. So the pressures for larger brains originated from complexity of social environments, where the others want to, swindle you into something or others that you need to compete with or cooperate with?
So someone gave a talk on the first day. I can't remember. You did. Yes. So you talked about birds. Oh, no, it's not part of things. But the idea was that if you have, so the relationship between large brains and different life history characteristics. So in birds, I've done this beautiful meta-analysis in which they showed that all the big brain birds have extended parental care, because you can't grow a big brain. You can only grow a big brain if you delay maturation. It's also a catch-22. So the big brain is really good once you've got it for solving problems, but you have to get there first. And that bridge needs to be overlapped by having other individuals caring for you and teaching you. So you basically have to have the ability to learn why it's so good to have a big brain. So I think the social brain, it's a correlation, and I don't think it's causation. So yes, big brain is important for having or being able to have all these social relationships. And in my story, of course, sociality was essential because you forge those bonds to protect yourself and reproduce. And I can't quite remember what his main point is with the social brain. Is it the number of individuals that you can have relationships with?
Yeah, I think so.
Yeah. It's done. Yeah, done as a number, the maximum number of people you have relationships with. But all these life history traits are so interconnected.
One more. I mean, this comes to Andy's point. I mean, surely the fact we're here discussing stuff today as human species or doing anything that's actually internal, our internal state in our mind and our instincts, why we would retain that brain, that vast brain, and that all the other species don't do it.
Yeah.
They're not discussing us.
Oh no.
Facing Andy, I guess. So, you know, is that sufficient? That sort of serious cognitive, and internal life and that expression of that, is that sufficient to keep going in a way that just doesn't occur in other species? And, that is now our sort of driving force.
Yeah, exactly. We're up here and we can't really go back. You know, it's one of those fitness peaks. You can only go back by going through a valley.
Yeah.
That's very difficult to do. So we're here. And we're going to have coffee.
Yes, we have. First, thank Madeleine again for wonderful